Okay. Hi, everyone. Welcome. Thanks for stopping in after lunch. So I'd like to talk to you about, it's actually two stories today. The first one is going to actually take off from the story you heard about from lake trout, and you're going to actually hear of a situation where we had overcompensation completely derailing an eradication program with European green crab. And the second one is going to be about what happens when we understand how synchronous populations of invasive species are and what may be behind them, and how that can help us target our management in the future. So I'll go take off here. Is this correct? Let's see. Nope. Let's try this. No. Oh, there you go. That's interesting. It's not on this, but it's on this. Cool. Oops, sorry. Okay, so as we all know, the best strategy for preventing invasions, for managing invasions is, of course, prevention. So we're all in on this. Of course, the marine folks are in on this with new VETA regulations and so forth. But occasionally, invasions get through. And in high-profile locations with high-profile species, we have to do something, whether it's eradication, containment, something. So today's talk will talk a bit about the circumstances under which eradication will or won't be successful. So this is an important issue for us, and as I will come back to, I will argue for avoiding eradication and choosing suppression, or what we call functional eradication. Now, to tie this to fishes, some of the best examples, although they're somewhat anecdotal, have shown the failure of aquatic species management. Smallmouth bass removal in a lake in upstate New York, after 7 years of diligently removing smallmouth bass, resulted in more smallmouth bass than they started with. So in cases where you have the removal of adults, you can either increase the resources available to juveniles, you can remove cannibalism, which is the mechanism that you'll see which happens with crabs. So you're losing adult control of reproduction and recruitment. Similar results were seen in a perch population following a disease die-off. So you can see these happening kind of repeatedly. And you would have thought that we might have learned some lessons. We haven't, and I think we're only now learning these. This is a paper by Zipkin a few years ago, sort of showing overcompensation. And let's see, I guess I can point to one of these. Whoa, not that one. Sorry. So what you see here is with adult, I guess I can only point to one, so I have to look over here. Let's see, I'm trying to more people over here. So what you can see here on this one is adult abundance and the number of juvenile abundance or recruitment. And what this means, overcompensation is a situation where, because of these kinds of density-dependent processes, adult control resources, cannibalism, adults at high densities control recruitment, or they keep it to a certain level. As you lose numbers of adults through harvest, through eradication, removal, you can have a situation where juvenile abundance can be very high. It's basically there's no control of recruitment. And it's this process that we unfortunately found out to be the case in our system. So the European green crab is one of the world's 100 worst invaders. It's introduced to 5 continental areas. There's been some substantial ecological impacts in Western North America, also substantial economic impacts, particularly on the east coast of the US. So we know it's a bad actor. It's had a long track record in lots of places. So we undertook a 5-year eradication program, and I'll thank Stephen for that. He's probably walked out because he didn't want to hear about this. But we've had funding from lots of places. But where we undertook a 5-year program to try to see if we could reduce or eradicate this species in a small embayment, which I'll show you in a second. And our data, as you'll see, suggested that this failed entirely because of that process I just showed you, overcompensation. And this is really the first example of this in a marine system, and probably the best example because we have a lot of different types of data which some of the lake systems don't have. So here you can see, and I'll use this one now, so we can go over here. And so we conducted this in a little embayment north of San Francisco called Seadrift Lagoon, just part of Bolinas Lagoon. And we undertook this eradication program really just as sort of a test of our ability to do this. We wanted to see if we could eradicate this given a certain amount of effort and time. This location is actually a very constrained, very small. It's about 2 kilometers, relatively isolated hydrologically. So it was a good place to go in and try to see if we can get rid of all of them. So what we did is we intensively trapped this for several years in a row. We used 15 collapsible traps several times a year, so enormous amounts of human effort to try to go get these things. And this is what the data look like. It's actually a really remarkable study. Yes, we're about ready to publish this now. So what you see here is this is 5 years of eradication. I'll come back to these in a minute or so. And this is the actual abundance. We actually have mark recapture data. This is actually catch per unit effort, but we actually have numbers here too. This is where we started with. So we went from this number down to less than 10% of the original population. So we're really successful. We were cheering. And then suddenly we had this enormous recruitment event. And we were back to where we started. What I'll point out, and one of the things I'd like to talk to today, which I didn't talk about 2 years ago when I was here, I'll have another story too, is that we've been able to maintain the population at about 30%. So the idea now is to keep the population at a level where we're getting ecological function, but we're able to sort of avoid this kind of reproductive event. Okay, so why do we think this happens? So one of the things that we can eliminate is just coastal, that was a good year for recruitment. So we have data from lots of other bays, all trapped at the same time under the same circumstances, and in none of these did we see any sort of similar event. So here you can see Seadrift, that was that big year. We see Tamales Bay, Bodega Harbor, all these other places, that's a different year. There's no recruitment, so there was no coast-wide event that contributed to a good year for green crabs. And that good year, of course, can be seen here. This is for Seadrift Lagoon. You can see the orange and green bars are basically the small ones, so you can see the population was almost entirely juveniles that year. Most of the other bays have significant amounts of adults, so this is what that looks like. So our data suggested, again, this was due to overcompensation. One of the key things in models of this is that if you have this adult control of juvenile survivorship, you can get this. And we suggested that it was due to cannibalism, because crabs are very likely to eat their own. In fact, we see this over and over. But one of the key parts was that we should not see cannibalism amongst the juveniles. If you've got this bay full of little crabs, if they start eating one another, that's not going to result in the kind of result we saw. So it should only be the situation where the adults are consuming the offspring. So we conducted a number of trials with this. So we did these 24-hour trials, lots of replicates with adults and juveniles of different sizes, and we gave them alternate prey in the tanks, and we repeated this 10 times in a row, so we did this over and over and over again to make sure we felt good about the results. We have lots of data for this study. And this is what this looks like. So basically, when the crabs are in different sizes, this means that it's a great big crab and lots of little crabs. These are when the crabs are all the same size. When they're the same size, the likelihood of predation is effectively 0. When the crabs are very different size, this is a logistic regression, it's almost certainty of cannibalism. Okay, so overcompensation, this kind of stuff also depends on recruitment being very linked to local population. In Flathead Lake with lake trout, of course, it's just one lake. We know that it's a contained population. With marine and coastal species, we can't be guaranteed of that, right? Because things are moving around. So we have to make sure that this recruitment event wasn't due to larvae coming in from other locations. So we had to see how close this was. So we conducted a genetic analysis to see if what happens, I'll point over here now, what happens in Seadrift stays in Seadrift. So we needed to do this genetically. So my colleague, Carolyn Teppold, who's worked along with many of us for quite a while, used mRNA from cardiac tissue and used SNPs and generated 5,000 or more than 5,000 mutual unlinked SNPs and analyzed this, looked at both allelic richness and generated some FST values. It turns out this Seadrift population, because of its isolated hydrography, is really distinct and really isolated. It was also the only site that had significant differences in pairwise FST, so I'll show you what that looks like. So here is the allelic diversity data, and you can see this is a map of the populations along the west coast that were part of this, so all the way from British Columbia down to Monterey Bay, and these are some of the sites in the middle, and you can see these pink spots. This is a principal component analysis. You can see the Seadrift population's way off by itself. So they're the pink ones. The nearest other bays are the red and yellow bays. So this is a very isolated bay So we assume that we going to conclude that what happens in Seadrift does stay in Seadrift and the population is really contained. So to finish this little bit, so eradication was basically a total disaster because of overcompensation. So what we'd like to sort of recommend, and I have a paper with colleague Steph Green in review here, is that we follow really what, say, the lake trout people have identified. You know, they said, let's go for 75% of the population. And what we would suggest is that that's what we should do also. In fact, we've been able to now maintain about 30% of that crab population. We have native species coming back in the system over time. So, of course, this requires a fair bit of work. Luckily, we have a citizen, what I call a community science program, because you don't have to be a citizen to participate, of course, but a community science program so that this will maintain the green crab population over time. So we've been able to sort of to see that through. So we would suggest that's the strategy, and to, with species like this, with certain fishes, crabs, avoid thoughts of eradication. Suppression or functional eradication is the way to go. Okay, so story number 2 is, well, how do we make sense of multiple populations along the Pacific coast? Because we'd like to think about whether they're isolated or not. We can undertake functional eradication if a population is isolated. But to what degree are populations, like with green crabs along the west coast, undergoing the same kinds of fluctuations because of external forces rather than larval dispersal and between them? So one of the processes that creates synchrony in populations is the Moran effect. So you can have spatially distributed populations that look synchronous or look on the same dynamics because of external things. In our case, we're going to think of oceanographic processes. This was first shown in lynx populations, sort of a standard lynx hair diagram here, but in fact, this was all due to just similar weather conditions. So what we're going to ask is, are the populations of green crabs along the west coast synchronous, and is this due to their own internal dynamics, larval dispersal, or just forcing from oceanographic processes? And I'll give it away, it's oceanographic processes. So this has been looked at in some other species. These are mussels from the outer coast. These are sites along the coast. These are years. And what you can see is there is some synchrony in the mussel populations. Blue years are bad years, red years are good years for them. So in some years, a lot of the populations are doing badly. Some of the other years are doing quite well. We also we published this a few years ago with native oysters, native Olympia oysters. Here, there's no synchrony. These are also populations along the west coast from BC down to California. And what you see is that in any one particular year, red years are bad years. In this case, green are good, but you see both red and green years in some years and red and green years. There's no good years and bad years for oysters. So we concluded that this is worth looking into with green crabs. They are much more influenced by oceanographic processes because they have, larval dispersal along the coast. So we decided we'd take a look at this for green crabs. Okay, the kinds of things that influence this, so I'm not going to spend half an hour on local oceanography. There are a few processes that influence coastal systems. This is the Pacific Decadal Oscillation, which gives warm and cold years along the whole west coast. This is ENSO, the famous El Nino Southern Oscillation. And suffice it to say, that gives sort of different processes to California versus the Northwest. Here you get kind of warm, wet winters. Here you get kind of drier winters. And then also upwelling, which is a very localized thing. It depends on the coast morphology and upwelling winds and things like that. So this is a much more local process. Okay, so what we did was, and I say we, I want to include lots of folks in this. So Tom Terrio, who's sitting there, Sylvia Yamada, Kat De Rivera, many people contributed this to data set. So this is very much a community of efforts. And they will all be authors on this, so sometime. So what you can see here is there's 12 populations sampled for 22 years, which I can assure you is a lot of data for a coastal invader like this. So this is what the data look like. You can see Washington, Oregon, and California down here. Red are bad years. Again, this is low populations. Green and yellow are better years. And what you can see is in some years, there's a lot of red up here in the Northwest. So they're definitely bad years and pretty consistent, whereas there's just not much pattern here in California. You can put all that data together in a single year. And what you see here is Green means correlation between bays. So these are these Oregon-Washington bays, very highly correlated in their population dynamics and recruitment, assuming. In California, not so much so. There's very little correlation amongst the California bays. So the Northwest is highly synchronous. California, the bays are very independent of one another. And this isn't due to distance, so we did a couple other correlations. So distance by bay, probably driven by the California populations. It means that bays that are closer to one another aren't necessarily more similar because of that difference that we saw. If we look at these different indices, the ENSO, PDO, and upwelling, we get different answers. So here's latitude. So this is California down here at 35, Washington, Oregon up at Washington at 45 degrees. And what you see here is the degree of correlation amongst bays at that particular latitude. So upwelling, which is actually stronger in California, actually sort of declines as you go from California to Washington. Again, over here, declining as you go from California to Washington. What you see here is no real effect of upwelling. A modest effect of PDO, so a little bit of effect here, but a much stronger effect and a very significant effect of El Nino Southern Oscillation. So here we get these wet, warmer winters, much drier winters up here, and you can see that has a big effect on the degree of correlation. The California bays are not correlated, once again. Washington bays are very correlated. Okay, so what do we make of all this? So one of the things that's driving this is El Nino results in this sort of pole-wide push of water from California northwards. You get these periods where you get a great sweep of warmer water with larvae, most likely, populating bays in Oregon and Washington. So it's these episodic recruitment events. Suddenly the Oregon-Washington bays all fill up with larvae in the same year. And that's what's synchronizing these bays. The California bays have recruitment of themselves each and every year. They're self-seeding. So those dynamics get out of phase, and they're basically independent, whereas the Oregon-Washington are basically reset during these El Nino events. Now the question is can we use some of this to understand dynamics? Well, we can, because we can say, well, with these El Nino events, we can predict when it's going to be a bad year for green crabs. When should you invest? If you're a shellfish grower, which are probably the folks that are most invested in this, when should they worry about checking their bags, dealing with green crabs? Well, it's these El Nino years, and it's also, you can look at the nearby bay and see how they're doing, and that's probably going to be a predictor of how you might be doing either that year or in the future. California bays, there won't be any predictor from this, so it's not going to help. Oceanographic processes won't help predictions there, nor your nearby bay. So we can, with some reasonable accuracy, predict when good and bad years will be, and then the location and how isolated bays will be. The last thing is that as green crabs move farther north to, say, they're already obviously in BC and they will be in southern Alaska at some point, is that these are going to be isolated populations. They're going to be by themselves and not experiencing much recruitment from the south areas probably for many years. So if you're looking to sort of do some sort of management, it's more likely to be successful and not be influenced by subsequent larval dispersal in the future. So I think what I'll do is I have a minute or so to stop. I just want to end with this by thanking several people in this. So for the overcompensation, Greg Ruiz, Kat De Rivera, and folks working with them, Linda, Andy, Julie, many others, and funding agencies. And for the Moran effect and the spatial correlation, of course, I'd like to thank Stephen and the Pacific States Marine Fisheries Commission that helped get this off the ground and has funded a lot of green crab work along the coast, National Science Foundation, Sea Grant, and others. So I think I'll stop there. Thanks.